Okay, everybody, we're going to do a very short talk about mechanisms of transport. This is going to be a very quick overview of things we've already learned. So we dug into each one of these things here in topics 1 through 8. Okay? So I handed out, and we're going to look at sort of an organizer for all these things here listed on this slide. So that organizer is something that I'll post on my site, on my Schoology site, for people who aren't in class today to have a look at. So we basically start with types of cell transport and divide it into active and passive. Let's start with passive transport, no energy, so write that next to your box, okay? We have simple diffusion, which is mainly osmosis. And osmosis can set up in a number of different ways. Isotonic, where concentrations are equal and water really doesn't have any net movement, so it might move back and forth, but concentrations will stay the same. A hypertonic solution has more things dissolved in it, and a hypotonic will have fewer, and these are relative to each other. If you have a membrane, one side will be hypertonic, the other side will be hypotonic. Okay, back up to passive transport, okay? Kind of following down here through facilitated diffusion. Remember, we talked about what a facilitator is, and we have two types of transport proteins. Channel proteins, which are just kind of tunnels, and carrier proteins, which sort of capture and allow a certain substance to come in. These are all, everything on this side is always high to low concentration. High to low concentration. All right, let's clear all this junk off, and we'll look at the other side here. Active transport requires energy, so you might want to write that down. And the main ways it happens are vesicles, where we pinch off part of the membrane. So draw maybe a little phospholipid, and just to remind you. And a cell will sort of wrap itself around something and engulf it, or the other way would be to sort of burp out a vesicle. So that happens with exo, where things are going out. So write out, write in. Okay, out for exo, write in for endo. For endocytosis, we can take things in for phago and pinno. So phago is usually eating something solid, and pinno is usually something fluid. So write solid and fluid on those. All right, so I gave you a little sheet to do some notes with. Can I go, or you need a sec? 1 sec, okay? So let me just try to out, in, eat, liquids.
Like the other side, that side all high, low.
Say that again.
Unlike the other side, this side all low to high, right?
This side over here is, well, it's usually low to high, but that doesn't really work with exo and endocytosis. It really is for the pumps. So the pumps go low to high. Good point. That's a good thing we can add. So you need to tell me, to summarize, okay, you need to be able to tell me types of passive transport, types of active transport, and you need to be able to outline them in an organizer. If you want to go watch a crash course on this to review, here's a link to it, but we're not going to do that right now. So that concludes lecture 2.9.